I very warmly welcome Professor Menno Witter from the Norwegian University of Science and Technology and the Catholique Institute for Systems Neuroscience. It's really an honor to have him here this afternoon, and it will be the utmost honor for our institute to take part in the awarding of a Doctor honoris causa later today. So the conference that Professor Menno Witter is about to deliver is actually the first physical conference of our institute since March 2020. So this makes having him here even more rewarding. Professor Bernard Ansel and Professor Pascal Hippocampal are the two godfathers or sponsors of this Doctor honoris causa. Their research, as well as that of many others, actually in our institute, as could be seen this morning during the workshop, have strongly inspired, so their work have been strongly inspired by the past and current work of Menno Witter. So, Pascal, may I ask you to introduce Professor Menno Witter, our distinguished speaker, who is also about to become a full member of our institution. Thank you.
Thank you, André. It's quite a lot of fun because we had to find out the name about what is our position, us as sponsor or godfathers. That in French is parrain, so we struggled a little bit and we decided for Godfather. Everybody thinks about the movie The Godfather, so I tried to find a Godfather outfit for that special event. Now you can start recording. Dear Menno, dear friends, dear colleagues, it's a pleasure, it's an honor, Menno, to welcome you here today and to introduce you for this conference just before the ceremony in which you will be awarded the title of Doctor honoris causa of the Université of Louvain. It's a pleasure, it's an honor, it's many other things. It is, however, anything but a surprise to see you here with us today. To me, it's quite just as if you were devoted to be here today. And I will try to illustrate it, just kind of if they are deterministic laws, just like in classical physics or classical mechanics, that would define your trajectory and that would explain precisely the reason why you are here. A little bit odd talking about physics, but this image is not taken at random in the way that, well, these laws of mechanics are telling us that, well, the trajectory of someone just like you, just other kind of people or other kind of object can be defined as soon as we know the initial positions and the forces that are driving the system. And as far as you're concerned, your initial position was probably in Amsterdam, where you got your master's degree in biology and your PhD in medicine. I will not tell about the date when you got these degrees because to avoid people to try to, by deduction, calculate your age. But I think that everything started there. The forces that guided you were, without any doubt, the will to know, to push the boundary of knowledge and to transmit it, as evidenced by the successive and increasingly important position that you got at the university to start in Amsterdam. PhD student and lecturer, assistant professor of anatomy and embryology, director of the Research Institute for Clinical Neuroscience, and the Interfaculty Research Institute for Neuroscience, director of the Graduate School of Neuroscience in Amsterdam, co-founding director and member of the board of the Center of Neurogenomic and Cognitive Research, always in Amsterdam. So driven, I suppose, by the same kind of forces, you joined as visiting professor of neuroscience, the Center for the Biology of Memory at the Norwegian University of Science and Technology in Trondheim in Norway. You are currently professor of neuroscience at the University of Trondheim and director of the Norwegian Research School of Neuroscience, affiliated also to the Kavli Center for Systems Neuroscience. You are also visiting professor of Tohoku University in Japan, visiting science scholar from Kavli Institute, Discovery Institute at Johns Hopkins in Baltimore, USA. From an academic point of view, Menno Witter is all over the world, I would say. You are also, or you were chair of the FENS Committee on Higher Education and Training, president of the board of the FENS Trust Foundation, Netherlands, and so on and so far. I will stop here, otherwise the conference will never start on time. That's very impressive, and it's impressive enough to make us all understand that you are a brilliant academic, but it's maybe not enough to explain or to specifically understand why you are here today in the health sector of UCLouvain. And there was an event that I would call a quantum event because it cannot be described by the classical laws of physics. That was an encounter, and it may not have happened. The probability that it happened was maybe low, but I think that the fact that we have met that day and discussed each other changed the state of our respective system, especially for us at UCLouvain, ever since. This day, can you remember it, Menno? It was on November 27, 2018 at 9 a.m. precisely. A delegation that was led by Wallonie-Bruxelles International, in which I participated, was welcome in your institute, the Catholique Center for System Neuroscience in Trondheim. The prestige of this institute is such that you were very much in demand, and maybe that your interest could be moderate or sometimes even lukewarm in welcoming all those people that come to you, to Trondheim. But something really happened that day, Menno. And the interest that you took in the subjects that we were presenting to you, the enlightened critical spirit with which you were able to discuss each of them, the benevolent authority that seems to emanate from your position that you occupy in this institute and among your peers, indicated that you were not only, we were not only dealing with a great academic, but on top of that, with an outstanding scientist. I can reassure you that it was not on that day that we discovered the name of Menno Witter, but it was simply the day we met Menno Witter. Just like in the TV series, it was the day that we saw him. For indeed, Menno, the aura of your work has made your name known in the community long before this trip to Norway for us. It would be very complex to attempt a synthesis and useless to illustrate it with metrics that are absolutely not the point in ceremony, just like today. Just one example. One of the core aspects of your research addresses the relationship between the architecture of neuronal networks and their function. You started a collaboration with the Kavli Institute in Trondheim and moved yourself to Norway more than 15 years ago. You started a collaboration with especially May Britt-Mother and Edward Mother at this Kavli Institute. It's not coincidence either. Expertise like yours in functional neuroanatomy is unique and in high demand. In particular, it made possible to position the cells in the brain that enables us to navigate in space, the famous GPS of the brain. Your contribution to the work rewarded by the Nobel Prize for Physiology and Medicine to May Britt-Mother, Edward Mother, and John O'Keefe in 2014 is simply major. It is a fact that for all of these reasons, you are here today, and I repeat it, to close the loop, it is a great honor for us to have you among us today. I will give you the floor just by quoting few words that are not mine. Life is haunted by breath, speaks with purest sense. When breath has departed hence, then science has eloquence. Please, Menno, the floor is yours.
This is quite an introduction to get started my lecture. Thank you very much. I really appreciate it, and I will use some time a little bit later after the ceremony to talk a little bit more, but I definitely remember that day that we met. And like it was for you, it also was for me an elevation to meet somebody like you, Pascal, who understood what it meant to try to do good science. And so you covered my CV, so essentially I can stop talking because the lecture that I prepared is really trying to put together the things that motivate me and how it helped me to get to the point where I am. So I really want to tell you about a structure in the brain that is called the entorhinal cortex. And this slide just illustrates this particular feature that no matter which species you would look at, these are all sections taken perpendicular to the long axis of this cortical structure. And there's two things that are striking. It's in most of the images associated with this structure over here that reminds you actually from the logo of this institute, because there's part of the hippocampus in that logo, and the hippocampus and the entorhinal cortex in all these species are very strongly associated. And the other thing is that the entorhinal cortex, and that's quite easily shown, for example, here, or for example here, has a bilaminar structure, and the cells close to the surface, to the outside of the brain, like for example here, those cells are really big. They're one of the largest neurons that we have in our brain. And that's true for any species that I've ever set eyes on. If you recognize one entorhinal cortex, you'll recognize them all. Which indicates something that's very interesting, because it's a very conserved structure. It's always there, it's always associated with the with the hippocampus. So this is a posterior view of the red brain, and here in this yellow color you see the hippocampus, and in the green and the yellow is the entorhinal cortex. As you can see, they're really closely associated. Now, of course, there's probably quite a few people with a clinical background who don't work with rats and mice, they work with humans. And this association of the two is true in humans as well. As you can see here, you see in red the hippocampus and in green the entorhinal cortex in the red. And if you simply move these two structures further ventral into the temporal lobe, you see the entorhinal cortex here and the hippocampus here. So they're still closely associated, they just have a slightly different position in the brain. Now, the entorhinal cortex and the hippocampus together build what we refer to as the brain memory system. You could be more specific, it's the episodic memory system. So it allows us to form memories and then to retrieve those memories actively, talk about them, and use them to make predictions for the future. What will happen if my computer crashes? How are we going to solve that? My brain started immediately to think like, what is going to happen? How do I deal with it? So what do we know about the structure? So in this cartoon you see a very schematic representation of the hippocampus. And what we know about the hippocampus is that there is a series of monosynaptic connections that run in one particular direction. The first one is the MASI fiber. The second one are autoassociative projections in a field that is referred to as the third cornea modus field. And then from there you have a massive projection onto the CA1 field. And then the next traditional input to that system originates in the entorhinal cortex, layer 3. And what you see here is the well-known trisynaptic circuit that people who started to work on the hippocampus, their first physiology, the discovery of the place cell by John O'Keefe, for which he got the Nobel Prize together with Edward and Maybrit Moser, was discovered based on this level of knowledge. Over the years, the system has become more complicated, which is fun for a scientist, but it also makes life a little bit more difficult because you have to take all these other parts into account. For example, there's an additional projection from the entorhinal cortex that arises from a different set of cells. It's layer 2 versus layer 3 that project to CA1 and subiculum. There are projections from CA1 to the subiculum, and there are both from CA1 and subiculum output projections to layer 5 of the entorhinal cortex. And finally, we now know that layer 2 and layer 3 of the entorhinal cortex are the main recipients of cortical information that is then processed by this loop through the hippocampus and then is terminated by way of layer 5 and returned to the cortex. So this is the basic scheme of what we currently understand of this system. Now for today, I want to focus essentially on these two components, the layer 2 and layer 5. And there's a number of reasons for that, and hopefully they will become apparent while I'm talking. And so one of the reasons to focus on layer 2 is, and I'll come back to that in the end of my presentation in a little bit more detail, is that entorhinal cortex layer 2 has a key prominent role to play in Alzheimer's disease. If you compare a healthy brain of the human with an Alzheimer brain, you see that there's a lot of cortical degeneration. This is the entorhinal cortex that starts to fall to pieces. Neurons die in this entorhinal cortex. This is a figure taken from a very well-known paper by Heiko and Evo Braak, who essentially paved the field of Alzheimer's disease in establishing a staging set of criteria, the Braak stages 1 to 6, which are still used in the clinic to describe the level of pathology and the associated levels of dementia. Now, interestingly, as you can see in this cartoon, is that there starts to be pathology in a particular cell layer, which is layer 2 of the entorhinal cortex. They form something that are called tangles, which is a deposit of hyperphosphorylated tau, which is one of the key pathological hallmarks of entorhinal cortex, together with the expression of amyloid plaques. Interestingly, this presentation, or this representation or expression of phosphorylated tau results in cell death. Particularly, this is a healthy human brain. These are clusters of layer 2 cells in entorhinal cortex. And if you look into more severe disease stages, these cells are disappearing. They're dying, and they're just disappearing from the network. And interestingly, it has been very well established, this is one example, but there's many papers, that show that if you look in mild cognitive impaired patients, and you look at the pathology in the entorhinal cortex by measuring the thickness in MRI, in structural MRI, that the ones who already start to have a diminished thickness of the entorhinal cortex are the ones that very likely will convert into Alzheimer's disease, and it's a very reliable predictor. So there is something about layer 2 cells that is intriguingly involved with the start of the disease. And it happens to be the case that we have been working on layer 2 cells for many, many years. And Pascal alluded to it, there are the grid cells, and I'll come back to that in a minute. So here you see the entorhinal cortex from a posterior view. And generally, the entorhinal cortex is divided in 2 subdivisions that are referred to as the medial, or MEC, and the lateral of LEC. I won't go into details. There is an endless debate about subdivisions in the brain and in the cortex, also in the entorhinal cortex. But this serves the property and the purpose of the talk. And these 2 areas can be identified using cytoarchitectonics. You can look at them in all kinds of functional criteria, chemical criteria. It looks as if none of the animations are working. Oh, there we go. They also show different projections into the hippocampus. And this picture that you see here looks exactly like the logo of your institute. The dark purple part of your institute logo has this pathway here coming in. And so this is what we refer to as the medial perforant path, and this is the lateral perforant path associated with these 2 structures, the 2 entorhinal subdivisions. And if you just notice in black, this is the distribution of the axon terminals from neurons in the entorhinal cortex layer 2. And these are the axon terminals of the layer 3 neurons. And if you just look at the green markers and you compare this one with this one, you see that these 2 blobs of terminals fields have moved together. So the point that I wanted to make is that the entorhinal cortex can be recognized, and it has specific differentially organized projections into the hippocampus. No reason for you to remember the details. I just want to make the point that these 2 areas can be recognized in the brain using a number of different methods and approaches. The interesting thing is that those 2 areas not only differ in terms of how they project to the hippocampus, they also differ functionally. In the medial entorhinal cortex, we have this firing pattern that has become famous because we got the Nobel Prize for it, which is referred to as the grid cell. So you're recording from a single cell in layer 2 of the medial entorhinal cortex. And each of these red blobs is a peak of firing of a single cell. And when we first discovered this, because we were looking for a kind of play cell outside of the hippocampus, we mentioned that, oh, this is a multi-peaked spatial cell. And then it took us a while to recognize that there's more to it, namely that these peaks are very precisely organized. They are always forming this equal-sided triangular or hexagonal shapes. So therefore, we started to refer to these cells as grid cells. In the lateral entorhinal cortex, there is no indication whatsoever, and there's many labs who try to record there, that you find these spatially modulated grid cells. What you do find, for example, are cells. So this is an example of the experiment that we do here and we do here. We have a box of about 1 meter or 1.5 meters. We let the animal running around, so in gray you see the walking trajectory of the animal in space, and we record from single cells. This is what you get in the medial entorhinal cortex layer 2. This is what you get in the lateral entorhinal cortex. The cell is absolutely silent. So initially we said, oh, there's something wrong with the recording system, we have to rebuild it, test it out, everything was fine. And then we put an object in this space, and then suddenly you see that there is a cell that likes the object. It starts to increase its firing rate. If we remove the object, the cell ceases to fire. But there's another cell type that doesn't really fire when we put the object there, but as soon as we remove it, it starts to fire as if it is telling the brain there used to be an object in this position. And actually, there's a very interesting story. By using these, what we call memory for object position cells, you can build a pattern, if you do it quickly enough, that looks like this. If you put the object in an hexagonal pattern, you build a beautiful grid cell, but it will disappear. Interestingly, these cells sometimes maintain a memory for the position of the object for about 13 days. So it's a very stable representation of there used to be an object here. The cell doesn't care about the type of object, but the fact that there is something there is important. So two functionally very different cell types. We've done a lot of work after the discovery of the grid cell. There's a ton of different cell types in the medial entorhinal cortex that you can see here. The important thing is that they're all spatially modulated. They're telling us something about space, and they're all based on intrinsically derived information. It's vestibular information, it's information about speed, orientation of the head. Again, it's a proprioceptive information system that strongly influences those cells. And a lot of those cells, these you see listed here, are there even if you deprive the animals from sensory input. So if you record from animals in the pitch dark, these cells will still fire. So they don't really depend on extrinsic sensory input. In the lateral entorhinal cortex, we now have very good evidence that these cells not only code for objects and objects in position, but they also seem to code for time in the meaning that they code for sequences during an episode. This lecture has been developing over a number of time. There were a number of events, and that sequence builds your full-blown episodic memory. Now let's start with the grid cells because we discovered them first. When we discovered about them, Edward and Maybrit came to me and said, can we look into the circuit? What is the circuit that underlies this very specific and peculiar firing pattern? And I just want to summarize, we did the study, I'll show you the results in a minute, but then immediately we started to look for what is the same network look like, what does the same network in LEC look like. If there's no grid cells, we had a very clear hypothesis that the network should be different. Because I tend to be a very strong believer, or I'm biased, that a network architecture will tell us something about function. Well, turns out you can be wrong. So there are two types of layer 2 cells. No matter whether you're in LEC or MEC, in this case they are green, and we refer to the green cells as Reelin cells because they express a protein Reelin. For now it's not important, it's just a protein that allows us to visualize the cells so that we can play with them. There's another cell type that is expressing calbindin, which is another protein. Again, it's not that relevant what it means, but it's a protein that we use as a marker. What you see, no matter if we look at mice, rat, or monkeys, there are green cells and magenta cells throughout both subdivisions, the lateral and the medial. These are the homologs in the monkey of these areas. And you see constantly that there are green cells, generally more superficially compared to the magenta cells. This seems to be the case in humans, although nobody has done a very elaborate study, but we know there are Reelin cells superficially in the human entorhinal cortex, and there are calbindin cells slightly deeper in the human entorhinal cortex. So we focused on these Reelin cells because those are the cells that project to the hippocampus, dentate gyrus, and CA3. And we had a very strong indication that those cells that express reelin are the grid cells. And I can come back to that if you want to know the precise arguments. So we started to record in ex vivo slices. So we cut slices of a living brain, we maintain them in a chamber, and we start to do patch recording. And this is work of Jay Cooey in the lab, who used to be a postdoc. And so if my cursor comes back, there are 4 cells here, number 1, 2, 3, 4. This is the permutation. All 4 cells are Reelin-positive stellate cells. And we asked the question, do these cells talk to each other, which is a very common phenomenon in cortical circuits. And the bottom line is, if you take 3 of those cells and you inject a depolarizing current, which means that the cell starts to fire, generate action potentials, and you record what happens in number 4, in this case, number 1, the only thing that you see is an inhibitory action or an inhibitory postsynaptic potential. And then you can permutate your recordings or your driving forces. You can drive 3 other cells and record from number 4. You can drive 3 others, record from number 4. And systematically what you find is that these cells don't talk to each other. They only result in an inhibitory signal. And that inhibitory signal is mediated by fast spiking parvalbumin positive neurons. So these cells don't like to talk to each other. It's like you're sitting next to your neighbor, you don't want to talk to him, you use another person that inhibits the 2 of you to talk. That's the network. The beauty of this network is that we could model it computationally, and it results in absolutely amazing stable grid cells. So we were very happy. That solves the issue. We know the network. Then we went to the lateral entorhinal cortex, predicting that there the network would be different. That turned out to be wrong. Here you have 4 Reelin-positive cells. They're fan cells, as we call them, and pyramidal cells. We do exactly the same experiments. We excite them, 3 of them, and we get an inhibitory response in number 4. So the baseline, the bottom line of this one slide is that the 2 networks in layer 2 of lateral and medial entorhinal cortex are similar. There are very subtle differences, but for now they look very similar. Still, the functional readout of the neurons that are wired up like this is completely different. And we can't get one to look like the other. We really tried, it's not possible. So then we asked the question, so what are these cells receiving? What is the kind of information that drives these cells? And so the interesting thing is that if you look at Reelin cells in the medial entorhinal cortex, they get, for example, input from the other side, the commissal input, and they get input from an area that is referred to as the pre- and parasubiculum, which gives a directional signal to the brain. And that's the input. So those cells, you can see it here as well in using physiology. This is a confocal experiment. You have the cell here, you have in yellow the commissal inputs, and in blue the inputs from the parasubiculum in this case. And you see beautiful synapses onto this dendrite. If you do an experiment in slices electrophysiologically, you drive the parasubiculum and the presubiculum, and a single cell responds with excitatory responses. So these cells have convergent cortical input. If you do a similar experiment in the lateral entorhinal cortex, so again, we are doing a patch recording in a slice, we're recording from Reelin-positive cells in layer 2 of the lateral entorhinal cortex, we find exactly the same thing, but it's different cortical inputs. In this case, I picked an example, the perirhinal cortex and the postrhinal cortex. Perirhinal cortex is very well established that it has something to do with object recognition and familiarity, novelty distinctions. And the postrhinal cortex is by now well established that it represents objects in a context. So it links a contextual information like I'm in this auditorium with what is happening in terms of spatial positions. So these cells, the fan cells, as you can see here in blue, receive an input in green from the postrhinal cortex, and we electrically drive the perirhinal cortex in this experiment. And what you can see is that no matter which cell type it is morphologically, as long as it is a Reelin-expressing neuron, they receive these convergent inputs. And in addition, we now have evidence that they receive olfactory input, they receive input from the medial entorhinal cortex, and from the opposite side of the brain, the contralateral entorhinal cortex, lateral entorhinal cortex. So again, irrespective of whether I'm in medial or lateral, these cells receive a lot of convergent sensory information in case of LEC, or intrinsically generated information in case of MEC. So this is the summary. 2 cell types in layer 2, wired up the same. They both receive convergent information from cortical areas, but the type of information differs. So then we ask, so can we do something else? What else do these cells talk to? Which is probably the local circuit. There's a lot of other neurons in deeper layers, so there's probably a lot of local circuits. And now the movie plays, great. So this is a movie that I really like. It's one of the first things that I did when I started to work in Japan in 1994. This recording was made in 1995. We're looking at a slice. Again, you have the hippocampus here. You have the entorhinal cortex here and a piece of cortex, the perirhinal cortex here. We have a stimulation electrode here in the dentate gyrus. And what you see in these warmer colors is the level of excitation or the levels of depolarization in the slide. So what you clearly see, you see a volley of activity coming out of this bipolar electrode to deep layers of entorhinal, superficial layers of entorhinal, and then to the cortex. So this output network actually functions, and it uses an intrinsic system where the deep layers of entorhinal cortex communicate with the superficial layers. So then we started to look more into detail in the deep layers, and layer 5 neurons come in 2 types. This is just an example. In both LEC and MEC, there's a cluster of cells in the deeper part that we refer to as 5B that stain for a particular transcription factor, in this case CTIP2. They also stain positive for PTP4. Again, it's a tool for us to target those cells, that we can recognize them. Layer 5A cells, layer 5A cells, both in LEC and MEC, don't seem to stain for this particular transcription factor, so we can use them, these factors, to manipulate the cells. These layer 5A neurons that don't express this CTIP2 or PCP4 are the ones that actually drive the output of the entorhinal cortex. So here you see experiments in mice, rats, and monkeys. In this case, we put a chemical tracer, which is taken up by axon terminals, transported back to the cell soma in the entorhinal cortex. And all the yellow cells, in this particular example, are cells that project to the nucleus accumbens. But we can do it for any cortical area, anything that receives output of the entorhinal cortex in these 3 species will label the same cells. And the yellow cells never, ever overlap with the blue ones that are expressing this marker, PCP4. So the 5A cells, as we refer to, are the output cells, and it's the 5B cells that receive the hippocampal input. So in order for the hippocampus to talk to the cortex, we need to get from 5B to 5A, otherwise we can't get out. And interestingly, we now know that these 5B cells do something very striking. Again, it's very similar between LEC and MEC. Here we label the cells in layer 5B in green, and the green means that we express a light-sensitive ion channel. That light-sensitive ion channel can be driven by light, by laser light, and then it will excite the axon terminal. So if we then shine UV light on these axon terminals in green, and we record from all these cells in magenta, from layer 5A, layer 3, layer 2, all the way down to superficial layer 2, you'll see that you have beautiful responses here. This is the number of cells. You have beautiful responses here, but the outliers are the 2 populations of Reelin cells. Reelin cells don't seem to get this hippocampal output feedback. Remember, the Reelin cells are the ones that project back into the hippocampus, dentate gyrus, and CA3. But again, the 2 networks in LEC and MEC are really similar. So this is the bottom line as summarized in the cartoon. You see the 5B cells, they receive hippocampal output, they project to 5A, can talk to the cortex, they project to layer 3 pyramidals and to layer 2B pyramidals, the calbinin cells, but they don't talk to these guys here in green that are the driving input to the hippocampal trisynaptic circuit. But again, there's no difference between LEC and MEC. So what on earth is causing this difference? I think that those are the differences in inputs, not the circuit intrinsically. Now, one final piece of the intrinsic circuit, and then I'll move on to the last part of the lecture. Entorhinal cortex the two areas talk to each other. So the lateral and the medial are not two isolated entities. They actually have massive intrinsic connections. And this shows it in a rat brain. If you put a anterograde tracer, which means it's taken up by the soma and it's transported to the axon terminal, you get a beautiful stripe of axon terminals here. If you move your injection site slightly more medial, the whole terminal band switches. If I would have moved it here, it would end up in this blue area. So there are bands of intrinsic connectivity that span the entire anterior-posterior axis of the entorhinal cortex. So they connect the medial with the lateral entorhinal cortex. And interestingly, exactly the same pattern has been found in monkeys, as you can see on the cartoon on the side. So what do these intrinsic connections mean? It's nice to know that there is a connection, so we wanted to see what do they functionally, what is the functional relevance of these connections. So, and as I mentioned before, these cells in the lateral entorhinal cortex that are here marked in this magenta kind of color are the Fen cells. We call them Fen cells because they're really amazing. For those of you who know Purkinje cells in the cerebellum, they have this massive dendritic tree in one plane. If you now take a Purkinje cell and you rotate it 360 degrees, you get a Fen cell. So a Fen cell is, if my head is the soma of a Fen cell, it doesn't have much dendrites on the basal side, but it has a dendritic tree on the top of my head that is around 900 microns in diameter, which is outrageously large. So these cells have a perfect capacity to collect information and then integrate it. And that's actually what they do, and I'll come back to that in a minute. So here we looked at the input from the perirhinal cortex and from the medial entorhinal cortex because I was intrigued by this intrinsic connection from medial to lateral. So what we do here is we electrically drive the perirhinal cortex. We express a light-sensitive channel here in orange in the inputs from the medial entorhinal cortex. And again, we record from Fen cells in the lateral entorhinal cortex. And so here you see the stimulus artifact of the perirhinal electrical stimulation. You see beautiful excitatory potential. Here we drive the light, and every time we turn on the light, you'll see a spike, a depolarizing current in the cell. So these cells integrate whatever comes out of the medial, which is probably spatial information based on intrinsic representations, together with perirhinal information that is representing object, novelties, familiarity, so information about objects. But if you look at this trace and you compare it with this trace, there is a striking difference. This trace shows a depolarizing current, and then it smoothly goes back to baseline, and that's it. All these traces go up, excitatory depolarizing current, and then you see a dip. It goes into hyperpolarizing currents, and then it returns to baseline. And if I blow it up a little bit, it's easier to see. So you see, in this case, the action potential coming up, or the depolarizing current, and then you have a hyperpolarizing current, and then it goes up again. So what is this hyperpolarizing current? And to cut a long story short, I'm not going to tell you all the details. This hyperpolarizing current is extremely powerful, but also extremely selective. So here we have a slice, and we record from layer 2 cells, in this case the Reelin Fen cells. We record from the deeper cells, the calbinin positive cells, and we record from layer 3 pyramidal cells, while we express a light-sensitive channel, just in the previous experiment, in the medial entorhinal input. So this is the cell type, we depolarize it, and we make all these cells burst continuously. Then we turn on the laser, and what you see here, this is a train of bursts because we depolarize the cell. We turn on the laser, the cell is dead silent. We turn it off, it gets back on. In the calbinin cells, nothing seemed to happen, and in the layer 3 cells, if anything, they actually increase their firing rate if we turn on the medial entorhinal input. So what it seems to indicate is that this projection from the medial entorhinal cortex can select how the lateral talks to the hippocampus. Because the layer 2 cells that are turned off are the cells that project the dentate CA3. And the layer 3 cells that are turned on are the ones that project the CA1 subiculum. So here we have an input out of the MEIL entorhinal cortex that selectively switches the pathways how the lateral entorhinal cortex communicates with the hippocampus. So, now what has this all to do with Alzheimer? I know that you're thinking about it, when this guy reaches the Alzheimer part. So this is what I think is so particular about layer 2 cells, as I showed you. They're Reelin positive cells. We know that those are the cells that express the tangles. Those are the cells that eventually die early on in the disease. And it particularly happens in the lateral entorhinal cortex. These neurons, the fan cells, have immense dendritic trees. They receive a lot of convergent information, and therefore they probably use an enormous amount of energy to do all these convergent calculations and to maintain this extensive dendritic tree. And finally, as was shown in a paper from the lab of one of my colleagues, Jamie Henry and Matt Nolan, they showed that these cells are absolutely critical for complex associations where you associate information about an object and the place of the object in the context. If you silence those cells, animals can still put an object in a place. They can still remember a context, but they can't make the conjunction between the three elements of an episodic memory. So I think that the cells in the lateral entorhinal cortex are the cells that provide the hippocampus with the complex set of information needed to form episodic memories. Now, what do we know about these cells? We reported a couple of years ago that these Reelin cells that you see here start to accumulate intracellular A-beta, soluble A-beta, in a model for Alzheimer's disease. This is a rat model in which a mutated form of the APP gene, so the precursor protein for amyloid, is expressed. And so it's only in these Reelin cells that we find this co-localization. Here you see it in LEC, here you see it in MEC. This shows the number of cells that express Reelin and the number of cells that express intracellular amyloid. So they perfectly overlap. This starts to occur almost in the second week after the animals are born. So the second week postnatally, you start seeing this increased expression of intracellular amyloid. You can say, well, this is a rat model, so big deal, but we found the similar co-localization in humans. This is not an extensive study. We looked at 3 pieces of brain of 3 different stages of Alzheimer's disease. But in all 3, we found that if we look at the number of cells, this is over 3,000 cells that we correlated and counted. Out of these 3,000 cells, almost all, 97.7%, express intracellular soluble amyloid. So both in early stages of Alzheimer, 1, 2, as well as in late stages. So the two seem to like each other, Reelin and amyloid, but liking doesn't mean much. It's just a correlation. But the interesting thing is that there's another correlation that starts to make it more interesting. So here we look at a 3-month-old rat and a 6-month-old rat. So again, it's a mutant. It expresses the mutated APP. And so here you have Reelin stain. And here we look at 2 different antibodies that stain intracellular amyloid. And this is the overlay. And if you look at the overlays, particularly in 6 months, you see if you, for example, look at this part here, the dots are Reelin positive molecules in the soma of the cell. And they coincide beautifully with the intracellular amyloid spicules in the cell. So it's not only the case that the 2 cells seem to express both proteins, they seem to actually be expressed in components of the cell. And these components we think has something to do with the lysosomal system and the autophagy pathway, but we're working on that. But we now know that the 2 molecules actually co-localize. We did proximity assays. But the most interesting thing, I think, that shows that the 2 are actually causally related is a paper that we just finished. It's on bioRxiv and it's under review in Alzheimer's and dementia, where if you start to look at the top panel, so again, this is a mutant model that expresses mutated human APP, so the amyloid precursor protein. We express a virus, and the green just tells you that these cells are infected by a virus, and this virus carries an antisense messenger RNA against Reelin. And you can see the result here. This is the control virus. These are 3 Reelin cells that are transfected by the virus, as you can see. And if you look in a slightly other area of the brain, the Reelin is gone. So we lower Reelin effectively, and this is the quantification. Here you see the manipulated virus that is expressing the microsense RNA, antisense RNA, and this is a control virus. And you see a shift in the population in the levels of Reelin. They're focused onto lower, much lower levels compared to the controls, if you look at the shuffled distributions. So lowering Reelin, and now look at this panel, results in lowering intracellular A-beta. And interestingly, it's not that it lowers intracellular A-beta because we do something to the APP gene, because in this panel, we did the same experiment, but now we're staining for AEP. So here you see APP in the cell, and here you see APP in the cell. So lowering Reelin has an effect on a splicing product of the APP molecule, not of the parent molecule. If I can get my cursor back, yeah, not in the parent molecule itself. So Reelin, manipulating the levels of Reelin, specifically influences the metabolism of APP, the splicing of APP. So that at least gets us closer to a causal relationship between the two. Now, this is the last piece of evidence that I want to show. This is unpublished data. We just presented them at the ADPD conference. This is work by Valentina Di Maria in Cliff Cantrell's lab. He's my neighbor in the corridor, and he is our genetic whiz kid. He makes all our viruses. So here we took the same animal. So again, it's this mutated APP-expressing animal. And now we inject a virus that expresses human normal tau. So it's not mutated tau, it's normal, healthy human tau. We transfect a bunch of neurons in the entorhinal cortex, in the lateral entorhinal cortex. You see there's neurons in layer 2. There's neurons in layer 3. The green tells us that they do express the virus and they do start to express human tau. We label the Reelin cells. And here you have the non-Reelin cells in the weaker stain. And interestingly, if you now look at a marker for phosphorylation of tau, in this case on the serine 396 position, you see that there is hyperphosphorylation occurring, but it occurs only in the Reelin positive cells, not outside. So, last part. How do we want to tangle this? This is all in vivo experiments. They're complicated. You need a lot of viral tools. So I just want to show you some of the recent tools, and then I'll stop. We decided to culture cells, and this is work done by Katrina Sjostad in the lab. We dissect out healthy adult layer 2 neurons or mutated layer 2 neurons. We culture them, and we culture them on a recording chamber so we can record from them. And we're now looking at the stability of the network, comparing mutated APP-expressing cells versus healthy cells. And then we can start injecting Reelin viruses to manipulate Reelin. We can start to manipulate the tau expression in those cultures. All the techniques are in place. But the real interesting thing, I think, is that we can not only do it here in a chamber, we can actually use this kind of structure that in a cartoon looks like this. This is work done by ECLII Hippocampal. It's a PhD student that we kidnapped from the nanoscience lab. He makes these multifluidic chambers. So there's 4 wells. We can culture different cell types in each of the wells, and the wells are connected by little microfluidic channels that allow axons to go in one direction. So we can have entorhinal cells in this chamber, dentate cells, CA3 cells, and CA1 cells. So we can build an artificial hippocampus. And this is proof, and this is work of Salome Nora, a master student in the lab, that shows that it works. So here we have a picture of healthy cells in LEC layer 2, the first chamber, healthy dentate granular cells in chamber number 2, healthy CA3 cells and healthy CA1 cells. And for now, these cultures are alive after 2 weeks of culturing. So we're just waiting how long they can live. And while we're doing it, we're recording. All these chambers have recording electrodes, and also the channels have recording electrodes. So we can follow the development of the network over time. If that works, we can then start to take one cell, one culture, like this one, and replace the healthy, the normal entorhinal cells with mutated entorhinal cells and see whether the network changes. And then we can use our viral tools to manipulate them. And this is work all done in the lab of Jan and Axel Sandvik, with whom I collaborate. I would not be able to do this myself. It's beyond my grasp how these techniques work, but they're extremely powerful and useful. So these cultures really allow us to grow Reelin expressing cells. So this is a control, this is just a number of cells in the culture. They're neurons, they express Louvain, and they express Reelin. So we have the right cell culture. And we can do the electrical recording. Here you see a spike of such a recording. As I said, we have viruses, and this is the last example. We have amazing viruses made by Stefan Blankvoort in the lab of Cliff Kentros together with Rajiv Nair. And so this is an example of a virus that was based on an enhancer approach that only labels Reelin cells in a wild-type animal. So we can take a wild-type animal, inject the virus, and do whatever you want. If we get the payload expressed in the virus, we can insert mutated APP, we can insert mutated Reelin, whatever. It's highly specific. This is for the medial entorhinal cortex. We had a pending patent. It was just approved, so now it's a patent to start manipulating towards treating Alzheimer's disease, if we are correct that it starts in layer 2 cells of the entorhinal cortex. Now, this is a virus that works in medial. Stefan just showed me the first slide, so this is really fresh, of a similar virus that works specifically for Reelin cells in the lateral entorhinal cortex. Last but not least, I talked about a lot, and I focused in the end on lateral entorhinal cortex that is relevant for Alzheimer's disease. So what about medial entorhinal cortex? Medial entorhinal cortex follows in the cascade of Alzheimer's disease, but it doesn't seem to be playing a role in initiating it. So again, this was a puzzle because we do all this work, and so what does it lead us to? And very interestingly, I was pointed out to a paper. I had never heard of this, but it's a real interesting paper. Vanessa Jane Hall sent it out because we did all this genetic screening. There is a disease which is called Sanfilippo syndrome type B. It's a kind of autism. It's a developmental disease. So these kids develop into young adulthood and then they die. They have severe autism and dementia and cognitive deficits. And the interesting thing is that if you look at the pathology in the brains of these patients, and so this is an animal model again, where the gene that is important for the syndrome is knocked out, is manipulated. So what you see is that in this knockout, and this is the control, this is heterozygous, here MEC looks pretty normal. LEC is absolutely normal. This is layer 2 of the layer entorhinal cortex. But now look here, you start to see the N-terminal A-beta expressed, there is APP expressed, and there is A-beta 140 and A-beta 142 expressed. Now, I don't know what this means, but I find it quite intriguing that in the medial entorhinal cortex that has a network that looks exactly like the lateral entorhinal cortex, it has the same cell types, it seems to respond in the same way to everything we do, but it doesn't seem to be involved in the initiation of Alzheimer's disease. But it seems to be involved in a disease that doesn't look at all at Alzheimer's disease clinically, but pathologically it has a lot of overlap. And then I'll just skip this. So I think that the difference in the phenotype of the cells in the entorhinal cortex really doesn't seem to depend on the intrinsic circuit, but it represents the difference in cortical inputs. MEC receives input that are internally derived that tells the animal where am I in space, can I position myself. LEC deals with information that tells me something about my outside world and changes in my external environment. So we think that the entorhinal cortex is actually a continuum. It's not two areas that do their own thing. It's a continuum that has a similar structure, and it just deals with different sets of information, and that decides what this part of the cortex is doing, which for me is still very counterintuitive, because I would say that a network tells us something. And I think that's true. I think that the network of the entorhinal cortex tells us something what the hippocampus wants to know. And they both deal with providing input to the hippocampus. But it doesn't tell us the phenotype of the two areas. So finally, I think that the networks in layer 2 are very, very similar, except for one point, which is plasticity. MEC is much less plastic than LEC. That has to do with the way the neurons are covered. And that might have to do something with the Sanfilippo syndrome. And I think that, and that's my current hypothesis, that difference in plasticity levels in layer 2 of the entorhinal cortex will determine the phenotype of the cell in terms of diseases. And with that, I would like to thank my lab, the people who do all the basic science, they're listed here, very active, all the people that are involved in the Alzheimer research, my technical staff, that are really very important for me. I can travel because I have an absolute fabulous lab manager, and I have very good technicians. I work a lot with Japanese colleagues. I have a visiting position at Tohoku University. Some previous members of the lab, the collaborations with the Sandvik lab and the Kentrels lab are very important. And of course, the people who pay for this and allow me to do this. Thank you. Thank you very much.